It's a true pleasure to be here. It's my first time visiting Vienna and first time visiting IMP, and I am truly excited to have this chance to visit both. And my interactions with colleagues at your institute has been very inspiring. I would like to say that we live truly in a special time. We have so many tools at hand that allow us to have the most precise information about our genetic makeup. We have the concept of how genetic makeups gave rise to disease and the traits. We can sequence our genome at $1,000 a shot and very quickly. So many people are starting to have their genome sequenced, to have their DNA mapped. It almost feels like we have unlimited capability to understand what life is about, and we even can potentially make changes on it. In the same time, we also felt helpless. There are so many diseases that we cannot cure, aging, related disease such as Alzheimer's disease, such as Parkinson, and cancer. Many of these diseases are inflicting huge burdens on society, on health, and affect many people that we know. At this moment, I don't know about you, I have always wondered, can we do anything about it with the knowledge that we have accumulated so far, with the tools in hand? And I would like to address some of that by focusing on a part of the human genome that have so far been, I would say, underexplored. And this is the so-called non-coding genome, which actually occupy 99% of the human DNA. And I'd like to talk about specific processes that actually now we are increasingly become aware playing an important role in the contributions to the phenotypes, contributions to disease, and potentially offer a window into both mechanistic understanding of the disease and trait and on development of new therapeutics. So that's about gene regulation in human trait and disease. The work really get attention of the society because with increasing amount of data about human genomes in the population, we now recognize that numerous sequences in the DNA, in our DNA, is conferring increased risk or increased propensity to give rise to traits and disease. And this is largely through genome-wide association analysis, where you can take the genome sequence of patient cohort and controls and perform statistic analysis to finding out regions with increased risk of disease. And so far, there have been over 100,000 such links made by the effort in the last 10 years since such study become popularized. The problem, though, is that the majority of such links or sequence variants fall within the regions that we call non-coding. 93% in a few years ago, a tally, and this number is actually also growing. The problem with this is that we don't know how to interpret the functions of such non-coding variants. A major hypothesis being put forward, and that is such non-coding variants in the non-coding region may confer risks to disease by altering transcriptional regulatory sequences and transcription of the target genes. So this is a simple diagram. Sequence polymorphisms or causal variants potentially disrupt or enhance transcription factors binding to the sequence that in turn would cause misregulation of the target genes and cause certain molecular phenotypes. They seem gain a lot of support in individual cases, but this has not really been systematically investigated. We have anecdotal evidence that this might be true, but is this generally applicable to the study of complex trait and human disease? To address this issue, we can investigate 3 predictions from this general hypothesis. The first prediction is if such a non-coding variants indeed disrupt transcriptional control, they probably are very highly enriched in so-called transcriptional regulatory sequences in the human genome, and also in a temporal or tissue-specific manner. Secondly, the target genes that predicted from such non-coding variants would also likely be enriched in certain molecular pathways that involved in disease pathogenesis. And thirdly, if such variants does exist and alter transcription factor binding, then you should find signatures that causal variants should affect the DNA binding of specific transcription factors. I like to address each of these predictions in the course of this talk. First, let's start with the first prediction. Is there enrichment of the sequence variants associated with disease that are transcriptional control sequences? To test this prediction, the major bottleneck we have is a lack of comprehensive knowledge of transcriptional control elements in the genome. Starting about 15 years ago, when we had human genome sequenced, NIH of the US government started an ambitious project known as ENCODE, as alluded by Alex. I was fortunate to be part of that program, and we started to develop technologies to map candidate regulatory sequences. These are hard to map based on sequence information alone because transcriptional control elements are typically very short and degenerate, hard to recognize using sequence information. But we instead recognize that they bear a lot of chromatin signatures. First of all, they are known to be accessible to DNAs, such as DNase 1 or nucleases, and therefore you can identify them based upon their heightened chromatin accessibility. Secondly, we and others also discovered that enhancers or promoters carries unique chromatin modification signatures. So promoters, where transcription initiates, associated with lysine 4 on histone 3 that are trimethylated, and as well as acylation on the histones, while enhancers, which instruct temporal and tissue-specific expression of genes, appear to be associated with monomethylated form of this lysine on histone H3. So this dichotomy allows us to separate candidate enhancers from candidate promoters. And they also bear hypo DNA methylation when they are active. About 10 years ago, with the advance of DNA sequencing technologies, we are able to combine biochemical assays such as CHIP chromatin immunopresentation with sequencing to develop tools to understand to map the location of such histone modification and determine the location of candidate regulatory sequence. So this methodology allowed we and others to identify a large number of candidate cis-regulatory elements. We call them candidate because these are sequences that look like control regulatory sequences, but we haven't functionally proved them. Regardless, accumulating evidence now suggests that a large fraction of the so-called candidate regulatory elements indeed can function as transcriptional control sequences. So far, about 20% of the human genome have been annotated as such candidate cis-regulatory elements through the work of ENCODE Epigenome Consortium and many investigators' effort that interrogated hundreds of human cell types or tissues. And a key result from such analysis is the recognition that distal control elements, such as enhancers, tend to be cell type specifically marked by histone modification. These are essentially the majority of the transcriptional control sequences breaking into 250 enhancer modules based on their patterns of tissue-specific activities. And each module is recognized to be specific to certain cell types. And what's remarkably, when this paper was published in 2015, is there's very strong enrichment of non-coding variants associated with traits and disease in each of these enhancer modules, just as predicted by this general hypothesis. For example, this is a section of this graph here. When you look at non-coding variants associated with traits such as Crohn's disease, type 1 diabetes, and so on, they're highly enriched, marked in this red color, in enhancers discovered in the T cell and related cell types, suggesting that indeed T cells might be involved in the initiation of such traits, and also confirming the prediction that transcriptional misregulation caused by non-coding variants could be the reason for such traits to arise. That was a few years ago, and one of the key weaknesses of such approach was the use of heterogeneous large bulk tissues, which lack the ability for resolution of cell types. Thanks to the development of single-cell technologies such as single-cell TAC-seq, now we have the ability actually to take a heterogeneous bulk tissue, resolving the individual subtypes of that complex tissue, and analyze the underlying transcriptional sequences in each cell type. So my lab adopted a technique called single-cell combinatorial barcoding and a TAC-seq strategy developed by Jay Shanduri's lab that allow us to interrogate chromatin accessibility in the individual cell level from almost 10,000 cells at a time. In each cell, we are able to generate over 5,000 fragments or potential or open chromatin in each nucleus. The general strategy of this is outlined here. It involves a combination of barcoding steps. The first step involves transposon TM5 mediated DNA barcoding in 96 well plates. Each well allows you to introduce a unique barcode to the open chromatin in that population of cells. You then mix all the cells from this 96 well together, redistribute into 384 wells, and add the second set of barcodes of 384 different barcodes. So the combination of DNA barcodes is very high, and if you sequence 10,000 cells, you essentially have ensured that 90% of the cells that you analyze are associated with unique barcode combination, which allows you to deconvolute the individual cellular chromatin accessibility in these maps. So with this type of tool comes the need to analyze the data. The single-cell chromatin accessibility maps from such data is still sparse. So we developed a tool that allows us to take the tens of thousands of individual profiles, process them, and group them into cell clusters based upon the similarity of open chromatin between cells. So for example, from a complex tissue of brain, we can actually resolve the underlying subcell types using a computational approach that we developed. And we can show that the chromatin accessibility derived cell types have remarkable similarity as if you perform the same analysis using single-cell RNA-seq data. And we can call the peaks of open chromatin from each subcell types and predict candidate regulatory elements in that cell types, and perform a series of subsequent analysis to give rise to the cell type specific control element maps. And this whole suite of software is named SnapAttack, and it's currently in GitHub for those of you who are interested in applying it. So as an illustration of the utility of such approach, I'm just going to describe to you how we use this approach to dissect the mouse brain. In collaboration with Joe Ecker and Holloway, Maga, Behrens from Salk, and also at Iran Makmal and Hongwei Dong from UCSD and the University of South California. What we did was we take mouse brain, we dissect from anterior to posterior into about 16 different slices. From each slice, we dissect individual brain regions for single-cell analysis. By total, we are looking at about 120 different brain regions. So far, we have already data for 45 such brain regions. They're coming from hippocampus, pallidum, striatum, olfactory areas, and the neocortex. From this, we collected roughly 800,000 single-cell chromatin accessibility profiles. And from that, we can get about 93 major and rare cell types mapped. From each cell type, we call the candidate regulatory sequences and combine them all together, predicted roughly 500,000 candidate cis-regulatory elements. Let me just briefly show you how this process took place. So for example, from the 800,000 single-cell chromatin profiles, we performed 3 levels of classification. On the top level, using snap attack, we grouped them into 6 different classes corresponding to non-neuronal cell types listed here, and 3 different types of excitatory neurons or classes, and then 2 different classes of inhibitory neurons. For each class, we perform additional clustering analysis using snap attack that led us to further identify a total of 44 major cell types. And for some of those cell types, we can perform even further subclustering. For example, here in one inhibitory neuron class, we were able to further subdivide them into additional groups. So altogether, we were able to identify 93 different subtypes that compose the mouse brain. And the percentage of each different classes of cell types are listed here. So majority of them are neuronal cell types, with about 1/3 coming from non-neurons. So this is a snapshot of the accessibility profiles for each of the major subtypes that we identified. And you can basically see that for marker genes of the major cell types, such as in excitatory neurons, not only promoters become accessible, and that's how we can identify such clusters, but also the gene body become more or less accessible. So using this profile, we can then annotate subtypes of the cell. And we can compare our annotation with those annotated before using single-cell RNA-seq, for example, from Stan Linnerson's lab from Karolinska Institute. And we found that from the 98 of the 265 cell types annotated using single-cell RNA-seq profiles, we were also able to recover using single-cell attack. The rest was not recovered in large part because we didn't cover the entire brain and also central nervous system. But this concordance at this stage already gave us a lot of confidence that we are able to identify both the corrected cell types and also the cis-regulatory element in each cell type. So with analysis of chromatin accessibility, therefore generation of the candidate regulatory element maps, we could then ask a few questions, such as cell type specific transcription programs. Again, using motif analysis coupled with predictions of essentiality of transcription factors, we were able to actually identify candidate transcription factors that play important roles for each of the neurons or non-neuronal cell types that we identified. So here, for example, in this excitatory neurons, we found several transcription factors in the BHL class and the neural D2 to be highly, to predict to have an essential function. And this is a class of inhibitory neurons, and we were able to predict that DLX family of transcription factors play important role here, and so on. So this general resource now will provide a roadmap for us to dissect really the composition of the complex brain and the circuits. Now, getting back to the question, are there enrichment of neuronal disease-associated SNPs in the brain cell type specific regulatory elements? To answer this question, we again take the mouse brain regulatory sequences, map them to the human genome. So basically, roughly 14% of the mouse genome now are predicted to be open and likely active in at least one of the brain cell types in the mouse. And of this, roughly 70% can be mapped to the human genome uniquely with various degree of conservation as listed here. And of those elements that map to the human DNA, we are able to perform enrichment analysis for sequence variants. This is a very complex map. It's not intended for you to read every line. What I'm showing you is basically, you can see that roughly this heat map breaking into two parts. So each row corresponds to a major cell type that we identified, and each column corresponds to traits and disease. And they roughly separate into two halves. On this half, these are mostly neurological traits, and some are metabolic traits. On the right-hand side, these are traits that are not really neurological, but again, may have a neurological disease traits. The blazing hot areas are, these correspond to schizophrenia, intelligence, and insomnia that have a very strong enrichment for a number of cell type specific enhancer elements that we predicted. If you look at this color, the red color represents excitatory neuron classes. So basically, there seems to be a preference for of enrichment for the schizophrenia-associated non-coding variants in candidate regulatory sequences discovered from excitatory neurons. And this is consistent with PsycENCODE publication, where excitatory neuron layer 2 and 3 have been shown to have a particular signature associated with schizophrenia. There are a few other classes of traits that also have association with enrichment. So what this map allows us to do is to identify not only the general excitatory neurons, inhibitory neurons that show such enrichment, but also pinpoint the subclass of cell types where the enrichment is the strongest, and then potentially provide perform follow-up studies. So from this, I hope to convince you that there is now a pathway towards mapping the regulatory sequences in a highly cell type specific manner across different body parts and tissues. And so far, the effort have led to the realization and the confirmation of this hypothesis that the candidate causal sequence variants are indeed enriched in regulatory sequences active in disease-relevant tissues. So not only this prediction is confirmed, but actually lead us to candidate tissues or cell types where the disease might actually be might be susceptible for the disease, and that's very exciting because a lot of complex trait, we know the DNA variants, but we don't know the mechanism. And having this first predictions of the cell type specific candidate regulatory elements, we actually now can make the first step of actually make some inroad into the mechanistic understanding. So how about the target genes of the regulatory elements? To test this, whether the target genes of these causal variants might be enriched for specific molecular pathways, we encountered a major bottleneck, and that is for most regulatory elements, they are far away from the genes they control, and it's not clear, at least from the sequence analysis, what genes they might be regulating. And the general hypothesis is that the distal element might regulate transcription by forming spatial proximity contacts. And this can take the form of loops, chromatin loops as shown here. It can take the form of just general chromatin folding and chromatin domains. Regardless, when an enhancer, for example, is engaged in regulation of the target genes, you should find at least temporary association of the sequence with the promoter of the target gene. Guided by such observation, we then took a step further. We asked, can we map such chromatin interactions in diverse cell types and use that information then predict target genes of the non-coding variants? We adopted a method developed by Joop Dekker's group called Hi-C. Many of you probably are familiar with this, that involves proximity ligation between distal DNA sequence element followed by high throughput DNA sequencing. And the result from such an experiment is shown up here as a heat map where the pixel color represent the frequency of ligation between two distal elements. Alex mentioned about this topologically associating domains. That's basically very visible from such maps. So this is the entire chromosome, but if you zoom into one segment, you basically see these blocks of DNA that show a lot of interactions among the DNA within a block, but not across the boundaries. And we define this as domains. And within each domain, if you sequence deep enough, you also begin to see off-diagonal dots that are signs of chromatin loops. But also, again, you can see not just off-diagonal dots, but also these close diagonal interactions that indicate further subdomains we call subtads. Using such maps, we can now begin to map in diverse tissues the interactions centered on promoters. A recent study from my lab essentially applied Hi-C techniques to a total of 27 human tissues. To save cost, we only sequence using capture probes interactions that are centered on 20,000 human promoters. And basically the results are shown in this type of maps where each arc represents a long range interactions that we consider as significant. So altogether, by studying 27 human tissues and cell types, which were previously analyzed by our group and others for epigenome maps, we now generate for the same tissues and cell types the Hi-C maps centered on promoters. And these maps led us to identify roughly 800,000 pairs of interactions between enhancer and promoters, or candidate enhancers, that correspond to roughly 400,000 unique pairs. Out of this, roughly 10% are between promoters, and roughly 90% are between a promoter and distal element. The interactions are highly cell type dependent. For example, this is an interaction that we mapped between a enhancer and a target genes that expressed exclusively in hippocampus, and the interaction frequency is much higher in the hippocampus than the rest of the tissues we examined. And indeed, when you identify such cell type specific interactions between enhancer and promoters, they somewhat correlate with cell type specifically expressed gene expression patterns for the same tissues. For example, this is showing correlation between histone modification of K27 acylation, which is an enhancer activity mark, and the promoter also. So basically, this each heat map, each box here shows very strong correlation between the distal enhancer and the promoter elements in that particular tissue. So when you see a diagonal indicating that there is a very strong correspondence between cell type specific chromatin interactions and cell type expression, the correlation in that cell specific cell types. This convinced, or at least gave us comfort that we can now use such maps to predict or infer candidate target genes for the non-coding variants. So by using the maps we generated and cross-reference the roughly 42,000 candidate non-coding SNPs from GWAS studies, we are able to identify or infer target genes for 27,000 of such non-coding variants. We now have performed a number of validation experiments. We tested roughly 13 of such predictions, and 70% of our predictions actually indeed are validatable using by doing CRISPR-mediated knockout experiments. I'm not going to show you that data because it's now recently published, but just to say that the predictions that we made indeed reveal known enhancer, known target genes of non-coding SNPs. In this case, for example, a obesity-associated SNPs, well studied. We correctly identified two of its target genes, IRX3 and IRX5. Then we ask, is there molecular pathways enriched for non-coding variants associated with certain human traits and disease? And to do that, we basically take the predicted target genes and we again perform a functional enrichment analysis. So first of all, let's look at Alzheimer's disease. This is, for example, the SNPs that we basically schematically SNPs and target genes. The target genes we predicted, many of which actually were already previously have been linked to Alzheimer's disease. So it's another validation of the utility of these maps. So we can then look for similarities between the predicted targets of different traits associated SNPs. So roughly 900 different GWAS traits, we were able to predict their target genes. We can then perform Pearson correlation analysis, grouping traits together if they share the same or similar set of target genes. And we reduce this 900 traits into roughly 40 clusters based upon the shared target genes. The 40 traits, some of the clusters are make sense. For example, the top here, we can see the cluster 5 include traits such as ferritin, serum iron, and these are related to blood-related traits. And this set of traits, cluster 38, include a larger set of traits, including cystic fibrosis, asthma, and even Parkinson's disease. The question is, does it make sense for them to be grouped together? Indeed, they are grouped together because they all share target genes that are enriched in the gene ontology, such as listed here, the peptide presentation via the MHC class, and also immune telomere organization, and then interferon gamma mediated signaling pathway. So in other words, the traits that are grouped into this cluster seem to all share molecular pathways involved in immune response. And this basically shows, again, for each of the clusters that we identified listed here, multiple traits, molecular pathways are enriched and therefore underlying the disease mechanisms of these traits. If you do the same analysis without this inferred target gene knowledge, you won't find this grouping structure, suggesting that this clustering approach indeed is driven by the predicted target genes of those non-coding traits. Again, so what this set of analysis allowed us to demonstrate is the following. That is, the candidate target genes of this non-coding variants in the genome are indeed enriched in specific molecular pathways. So such knowledge, again, generate hypothesis for us then to pursue the mechanisms of such traits. So again, out of the three predictions, we now have evidence that two of which are indeed confirmed. And now let's look at this final prediction. Do the non-coding variants associated with different traits tend to disrupt transcription factor binding? I think this is this, although makes sense, but it has been so far hard to validate or confirm. Because we don't have really complete knowledge of how transcription factors interact with sequences, especially how they distinguish between different non-coding, different alleles of a SNP, for example. In the past, we typically use a way to represent the DNA binding of the transcription factor. This is called position weight matrix, here represented in this logo that you are potentially all familiar with. The height of each base represents the frequency that base is recognized by a transcription factor. And if you want to predict whether this factor is affected by a DNA base here, you apply this matrix and you make predictions. And the differential prediction, for example, here represent the frequency, how you can compute a score for each sequence, and that score, the differential of that score represent the differential binding. The question is, does this differential score reflect differential transcription factor binding? This actually hasn't been systematically investigated, and this is something that we have now interrogated using experimental means. We formed a collaboration with UC Pathology at Karolinska Institute to adopt the so-called Silex assays to interrogate transcription factor binding in a very high throughput manner. The Silex stands for Systematic Evolution of Ligands by Exponential Enrichment. So this approach involves incubation of a pool or highly complex pool of DNA oligos with recombinant transcription factor. So the bonded DNA on the beads can be washed, and the bonded DNA is eluted and amplified, and then used as input for a second round of binding and elution. This experiment is repeated multiple times. With each round of binding and elution, you expect that the true substrate of a transcription factor to show increasing abundance or increasing enrichment along these cycles of Silex experiment. We designed this oligo library to contain essentially a 40 base pair long DNA oligo. For each SNP, we design a pair of such oligos with one corresponding to the reference DNA, another correspond to the alternative alleles. What you expect then is that if there is a differential binding between this reference and alternative allele, you would expect to see that the one allele to be highly enriched while the other allele to be somewhat depleted. So we use two scores to represent the binding of the transcription factor and the differential binding of the transcription factor among the two alleles. And the first score is called oligo binding score. And this oligo binding score, if you do the, actually we derive that, has a linear relationship with the log transformed binding constant between the transcription factor and the DNA. And by analogy, this PBS score, the preferential binding score, essentially is the area between these two curves, also have a linear relationship between the differential of the binding constant of the transcription factor to these two different alleles. The beauty of this experiment is that you can perform it in an extremely high throughput manner. So we performed such experiment using 750 assays corresponding to roughly 270 distinct transcription factors after the experiments and the QC. And basically, we were able to identify 11,000 candidate SNPs. From the initial pool of 95,000 such SNP, we found roughly 10% of them to show differential binding to at least one of the 270 transcription factors. And now we have the data to evaluate how transcription factor binding can be predicted by PWMs. And to test that, we use an approach called precision recall, and where the recall represents how many of the elements can you recover. This essentially out of the differential binding alleles for each transcription factor, we basically plot this graph, and the precision represents how precise your PWM predictions are. So in this graph, if you have a plot shows this curve here with underlying areas approximating 1, that basically indicate very high prediction power. But if you have, for example, this type of curve, that means it almost has 0 prediction power. So we performed this kind of analysis for each of the about 161 transcription factors for which we have enough data to perform such analysis. And these areas under the precision range from 0 to 1, with some of the factors are showing very high predictability by the PWMs, but the majority of the PWMs have a poor and very poor predictive power. Some are even poorer than a random guess. So this basically indicates that the PWM matrix that we have, even though it can predict where transcription factor binds in terms of patterns, but lacks in general the predictive power of distinguishing between the two alleles. How do we know that our experimental measurements are more trustworthy than the PWMs that have been derived previously? We have multiple lines of evidence to support that, one of which is shown here using star-seq method, which developed in Alex's lab many years ago. So in this approach, we take the oligos representing different SNPs, clone them into a reporter library, basically on the 3' end of the reporter, and we then transfect them into cells and measure the abundance of DNA and RNA barcodes. So basically here represent how the RNA molecule expression from some of the oligos can be identified here as red. Then we can take this active DNA candidate enhancer oligos, and we ask, do they show allele-specific activities? Again, by comparing the two different alleles in this data point, we are able to identify a number of SNPs, roughly 500 of which are showing such differential enhancer activities. Then we can now begin to ask which one predict better? Is it PWM predict better of this differential enhancer activities of the two alleles, or the in vitro Silex experiment? And the answer is the Silex experiment predicted preferential DNA binding track much better the differential reporter activities of each element than the PWMs. Here, basically, we rank this roughly 500 different allele-specific SNPs into 5 quantiles based upon high to low. So the top quantile obviously show the strongest differential prime enhancer activity, also show the strongest enrichment of preferential transcription factor binding as predicted by the Silex experiment. And as the quantile go lower and lower, which represent to lower or no differential reporter activities, this predicted differential binding by the Silex experiment also drop. And you don't see that trend if you use the position weight matrix. So this suggests that the in vitro binding data that we collected are more predictive of the activities, at least, of transcription factor. And we also have other data, which I won't show, indicating that ChIP-seq data is also better predictive by the in vitro Silex data than the PWMs. Now let's test the final prediction. And in this case, the prediction is whether the causal SNPs for complex trait are indeed enriched for transcription factor disrupting SNPs. We focus on type 2 diabetes in this case. Number one is because this is a very prevalent complex trait, and there has now been very careful and detailed analysis of non-coding of SNPs associated with type 2 diabetes. Roughly 150 such loci have been linked to the risks of type 2 diabetes, and they provide a roadmap for us to understand the mechanisms of this trait. A few years ago, Mark McCarthy lab from Oxford have performed the fine mapping of such type 2 diabetes by interrogating roughly 500,000 individuals from multiple cohorts. And from this map, you can identify candidate causal variants based upon a calculation here using this one called posterior probability assessment. Essentially, the higher this number, more likely this is causing, this is the true causal SNPs. And the lower this number, the less likely it is. So we would expect to see that as the likelihood the SNP is a causal SNP, it will show enrichment for transcription factor binding disrupting SNPs. And indeed, that's the case. So basically, when we say PB SNPs here, I show you basically these are derived from in vitro Silex experiments. There's increasing amount of enrichment as you basically see the levels or likelihood of that set of SNPs are causal. But if you do the same analysis only using PWM predictions, there's no such actually enrichment. In fact, there's even a decrease. This definitely shows that the PWM is not that effective tool for us to understand the function of the causal variants, but rather the in vitro binding data that we empirically generate are actually much more useful. We can actually take one such predictions and show that it led us to a intended, basically here is a SNP. We predict that it's disrupting transcription factor binding of CBP beta. And we test this by essentially using CRISPR-I to shut down this enhancer in the HepG2 cells where CBP beta is actively highly expressed. And indeed, the intended target genes inferred by high Cmaps are reduced. But when we do the same test in a different cell type, HAC293, where the CBP alpha beta is not expressed, the target genes is not affected by this CRISPR-I experiment. So this assay showed how we can now take the SNPs combined with the predicted disruption of transcription factor binding to identify the candidate transcription factors affected by the binding, and also use this to infer the likely causal variants of the transcription factor traits. So I just conclude, I provided evidence for 3 predictions of the general hypothesis that non-coding SNPs affect transcription factor binding to disrupt transcription. And the first is we showed that the likely causal SNPs are enriched in the candidate cis-regulatory elements in specific tissues and cell types that are relevant to the disease. I showed that potential targets of the non-coding SNPs are enriched for specific molecular pathways, and also for at least type 2 diabetes, the likely causal SNPs are enriched for DNA binding disrupting variants for specific transcription factors. So this provide a general roadmap for us to now tackle the human traits and disease. Starting from the GWAS SNPs, we can now make predictions of their target genes and identify the molecular pathways affected by such non-coding variants. And use such information, hopefully we can now develop better understanding of the disease, develop more accurate diagnostics and therapeutics. There remains a lot to be done. We still need to generate more comprehensive maps of candidate regulatory elements. And we also need to have a better understanding and catalog of transcription factor binding to DNA. Our studies shows how important it is to actually empirically determine the transcription factor binding against the different sequence variants. And also, we are still at the early stage, but we need to have a more accurate predictions of the target genes of each candidate cis-regulatory elements. In conclusion, I'd like to thank my colleagues for each of the study. The first part, I in particular thank Sebastian Prizo, a former postdoctoral member who set up the single-cell attack-seq from in my lab, and Rongxing Fang, a former graduate student who developed SNAP attack, and Yang Li, a current postdoctoral fellow who is involved in brain maps. And the targets predictions using high-C maps were carried out by a former graduate student, Anthony Smith, and a former postdoctoral fellow, Yingqiang Zhang and Yarui Diao. And the more recent TF binding Silex experiments conducted by 3 former lab members, Jian Yan, who did the experiments together with UC Tapali, and Yun Jiang, who analyzed the data with the help of Andre Santos. And we are very grateful for our collaborators and funding agencies listed here. And I'm very glad to take further questions if there is any. Thank you.